afternoon, dear students. Myself, Dr. P. Samuel, Assistant Professor, Department of Biotechnology, Bishop Weber College, Trichy. In this video tutorial, we'll be going to discuss about water-borne diseases with special reference to cholera. So, what is cholera? Cholera is a disease caused by the bacterium Vibrio cholerae. The same bacterium was once considered as a dangerous pathogen because before 50s or before 60s, there were a lot of news regarding the outbreak of cholera among the community of people who don't have any proper access for water. So, in such cases, cholera was a great headache to the people in those days. But now, due to the development of technology, right, we have eradicated cholera. And as I told you, it is caused by the bacterium called Vibrio cholerae, and it seems to spread among the people through consumption of contaminated water from sources like river, lakes, and wells, right? So as the term describes, it is a waterborne infection, which means the pathogen spread among the individuals through the consumption of contaminated water, right? So this is the first point you have to remember. Second thing about the pathogen, right? So Vibrio cholerae is a gram negative bacterium. What is gram negative bacterium? It is actually a staining procedure used in microbiological laboratories and other units to distinguish gram positive and gram negative bacteria. This staining works based on the composition of bacterial cell wall, right? So this particular bacterium called Vibrio cholerae, it's a gram negative bacteria, and it has uh, what's that? It lacks a thick peptidoglycan layer which is found in gram positive bacteria, right? And the thin layer, the thin peptidoglycan layer of this bacterium is located between inner and outer membrane of the cell. And the outer membrane of this particular bacterium called Vibrio cholerae is characterized by the presence of lipopolysaccharides, which is shortly represented as LPS. And this is the important virulence factor of the bacterium, right? And its shape is very special. It is a comma shaped bacterium, uh, and it has flagella, right? So it is motile in nature. The organism is motile, and the movement of this bacterium is facilitated by the presence of a polar flagellum that allow the bacteria to swim through the water, right? So this is the characteristic features. So in the first slide we studied about what is cholera. In the second slide, in the present slide, we came to know that it is a gram negative bacterium. The major virulence factor of the bacterium is lipopolysaccharide, which is present in this bacteria, and it is also responsible for the endotoxic properties of the cell. It is a motile bacterium. The motility is facilitated by the presence of polar flagellum. The polar flagellum allow the bacteria to swim across the environment. Right. Let's move on to the next slide. How this bacterium enters into the body and spread within the human body. Right. How it enters and how it spread within human body. As in the previous slide, I mean in the first slide, I told you that this bacterium enters into the human body through the consumption of contaminated water. Right? Remember, recall that particular point. So it enters into the human body through a process called ingestion. So when we take contaminated water, contaminated food or water, the bacteria present in the water enters into the human body, right? As soon as it enters the stomach through duodenum, it enters into the small intestine. The retention time of water when compared to solid food is very very low. So, as soon as the water enters into the stomach through duodenum, it enters into the small intestine where the organism is getting colonized on the epithelial cells. Epithelial cells of the small intestine, and how this particular bacterium is able to bind on the epithelial cells of the intestine. So, this may be a question, but the answer is in the statement itself. It attaches to the intestine through adhesins and pili, right? So, these are all the structures which are adding virulent nature to the bacterium. So after attachment, it started to colonize and it produced a toxin called choleragen, choleragen or cholera toxin. And the protein, it is proteinaceous in nature. It binds to small intestinal cells and activates adenylate cyclase enzyme. So this results in the increase in cyclic adenosine monophosphate, right? So it enters into the body through consumption of contaminated water. So from the stomach, it escapes into the small intestine. There it attaches to the epithelial cells of intestinal lining. After attachment, it becomes colonized on the surface of cells by using the virulence factors such as adhesins and pili. After attachment, it produces a protein called adenylate cyclase, which enhance the concentration of cyclic adenosine monophosphate in the cell. Right. This AMP, cyclic AMP level trigger an efflux of fluids. So as the concentration of what's that cyclic AMP increases, it triggers the efflux. Efflux means removal of water and essential ions or electrolytes from the intestinal cells into the lumen. Right. This causes watery diarrhea, and the excessive loss of water leads to severe dehydration. The very notable symptom of the cholera is the rice water stool. The excreta of the infected patient profuse stool which resembles the texture of rice water, right? Remember. And how it is diagnosed in the laboratory. First one is stool culture, right? So the sample is collected from the patient and it is cultured on selective media called alkaline peptone water, right? Alkaline peptone water or thiosulfate, citrate, bile salt, sucrose agar, which is called as TCBS agar, right? So these nutrient medium support the growth of Vibrio cholerae, and which is further identified by carrying out various biochemical procedures. And microscopic study, as we discussed in the second slide, it is a gram negative bacteria. It is motile in nature, and it is comma shaped in shape, so that we can identify all these things through microscopic analysis. And a rapid diagnostic test is also available in the laboratory, which helps to identify the pathogen present in the stool samples. And molecular techniques such as PCR, right, advanced studies like PCR, can also enable the laboratory technician to uh, pinpoint the pathogen in a short span of time, and how it can be treated and how it can be prevented. So treatment can be done by giving immediate rehydration, because the characteristic feature of cholera is the profuse diarrhea, right? Continuous diarrhea, which leads to severe dehydration in patients. So the patients can be stabilized by giving rehydration procedures. Rehydration procedure in the sense giving solution which is rich in electrolytes and other essential ions for the body. And in severe cases, intravenous fluid replacement may also be given. Sometimes antibiotic therapy like doxycycline, azithromycin can also given, right? And zinc supplementation may also be helpful. Zinc supplementation, and how it can be prevented. So the only thing, so during rainy days, we can hear warning from the government side, right? So people, uh, before the consumption of water, people should boil it, right? So the water can be boiled enough so that it prevents the growth of vegetative Vibrio cholerae in the water. This is a well defined mean of preventing the spread of cholera pathogen. Vaccination against cholera is available and recommended for travelers to areas where cholera is endemic. Once we detect the pathogen on an early stage, it will help us to reduce the rate. Mortality rate. So the disease is preventable, right? But immediate care should be taken, right, by the individual, right? So immediate care in the sense, once he or she is experiencing the symptom means he or she should go to the physician immediately and start summarizing or start explaining the experiences to the physician for the correct kind of treatment. Hope you will understand what is cholera and the structural features of the pathogen and the pathogenesis of cholera disease and how it can be prevented and treated. So we'll meet in the next video lesson. Till then, it's bye from Dr. Samuel. Bye bye.